Hello, welcome to my YouTube channel. And today we will learn about complement system, which is an important topic of immunology. आज हम लोग देखेंगे complement system के बारे में, जो कि immunology का एक important topic है. complement क्या होता है? complement ये complement it is a group of protein normally present in serum in an inactive form. Means complement जो होता है एक ग्रुप ऑफ प्रोटीन्स का ग्रुप होता है जो कि सीरम में प्रेजेंट होते हैं इनएक्टिव फॉर्म में. बट व्हेन इट गेट एक्टिवेटेड ऑगमेंट द इम्यून रिस्पॉन्स. मतलब जो ये इनएक्टिव प्रोटीन्स होते हैं जब एक्टिवेट होते हैं तो इम्यून रिस्पॉन्स को जनरेट करते हैं या इम्यून रिस्पॉन्स को बढ़ा देते हैं. complement system constitute about 5% of normal serum protein. Level does not change after infection or vaccination. Comprises of 30 serum protein. complement जो प्रोटीन होते हैं वो टोटल लगभग 30 टाइप के होते हैं. 30 टाइप के सीरम प्रोटीन, जिसको ये 30 टाइप के सीरम प्रोटीन हैं कॉम्प्लीमेंट, उनको 3 कैटेगरी में डिवाइड किया गया है. कॉम्प्लीमेंट कंपोनेंट, प्रोपर्टीन सिस्टम, रेगुलेटर प्रोटीन्स. कॉम्प्लीमेंट कंपोनेंट इसके नाम 9 कंपोनेंट है, 9 कंपोनेंट है. C1 to C9, C1Q, C1R and C1 is 3 subunit of C1 complement. Liver is a major site for synthesis of complement protein. Other sites are blood monocyte, tissue macrophage, epithelial cell, GIT and genitourinary tract. Now we will see about complement pathway. There are 3 types of complement pathway. Classical pathway, antibody dependent, triggered by the antigen antibody complex formation. Second one is alternate pathway, antibody independent pathway, triggered by the antigen directly. And lactic pathway resembles classical pathway but antibody independent. Stages of complement activation. There are 4 main stages in the activation of any complement pathway. Initiation of the pathway, formation of C3 convertase, formation of C5 convertase, formation of membrane attack complex. Characteristic of complement system is bind to Fc region of antibody and help in fixing or consumption. Role of antigen can only fix those antibodies that bind with antigen. Means complement function of complement depend mainly on nature of antigen in case of classical complement pathway. In other pathway it also depend nature of antibody species non-specific present in sera of all level. Efficiency decrease with an increase in taxonomic group. Means if close taxonomic group is there, then chances of complement matching is complement will be very close and very similar. These are complement protein are heat level and get denatured by heating the serum at 56 degrees centigrade for 30 minutes. And after this 56 degrees centigrade at 30 minutes heating, it called inactivated serum. When complement system get inactivated by heating, then serum called inactivated serum. Complement activation synthesized in inactive form like zymogen. Zymogen is a proteins which synthesize in inactive form and activated by proteolysis have two fragment, larger fragment designated as B fragment, smaller fragment designated as A fragment. Exception is in case of C2 complement where C2A is larger fragment. Larger fragment participate in the cascade reaction of complement pathway. Fragment of complement interact in definite sequential manner, that's so called as cascade reaction. Cascade reaction is here complement protein react one after other. This is all the three pathway of classical complement pathway. Here a classical pathway where antigen antibody complex formation, this is the initiator of this pathway. Antigen antibody complex, then complement protein C1q, C1q, C1R and C1S comes get activated in presence of this calcium ion. Antigen antibody complex bind with C1 complement, activated C1 complement, then C4 get activated and C4 act on this antigen antibody complex which bound with C1 complement. C4A is a byproduct which is N-phylatoxin. C4B get bound with this, then resultant form is C14B bar. Then in presence of magnesium ion, C2 complex comes and C2 break into part C2B. C2B is have kinase-like activity and C2A get bind with this complement and form the C3 convertase. C3 convertase further act on C3. This is the junction point where all the pathway get come to similar point. Like here C3 convertase form in lectin pathway. Membrane binding lectin when combined with mannose residue on microbial surface, like membrane associated serum protein. This complex formation takes place and further C4 comes in contact and C4 convert into C4A and C4B. C4B bind with this complex and this here is formed MC4B bar. C2 comes there and break into part C2B and C2A. C2A get bind with this complex and form C3 convertase. And in this alternate pathway C3, here's C3 complement spontaneously get hydrolyzed and convert into fragment C3A which as byproduct and remove activated C3B bind with initiators like endotoxin, then form C3B bound formation. C3B bond form react with factor B. Bind with factor B is it forms C3B capital B bar, and factor D comes and it work B to BA and BB. BB is react with this complex and C3B capital B small b bar. This is C3 convertage of alternate pathway. C3 convertage pathway of alternate pathway. C3 convertage of alternate pathway is also called properding stabilizer. Now C3 convertage of all the pathway. Convert C3 to C3A and C3B. C3A is anaphylaxis and C3B bind with C3 convertage, which is here and here. Now C5, this is C5 convertage, which is equals to C3 convertage plus C3B. C5 convertage convert C5 to C5A and C5B. C5A is anaphylaxis and C5B bar further react with complement C6, C7 and C5, C6, 7 bar further react with C8 and 9 and form membrane attack complex C5B 6, 7, 8, 9 bar. This membrane attack complex cause cell damage and formation of pore in the cell. This membrane attack complex lead to formation of pore or channel in the target cell, which lead to which permit ion in the cytoplasm of target cell and cause cell damage or lysis. Now we will see about difference between all the three complement pathways. This is summary of this detailed mechanism. What happened here? I will summarize this. What is actually here? Formation, how it goes on. Classical pathway, alternate pathway, and lacking pathway. In classical pathway, activator or initiator is antigen antibody complex. Means antigen antibody complex whenever formed, then it activate classical pathway of complement. Alternate pathway, endotoxin IgA, IgD antibody or cobra-venom nephritic factor is initiator in case of alternate pathway. These initiators bind with activated C3B complement and formation of C3B bounded with this initiator complex. In lactin pathway, activator is carbohydrate residue of bacterial cell wall, which may be mannose binding protein that bind to boost lactin antigen. First complement activated in case of classical pathway is C1. In alternate pathway is C3B. In case of lactin pathway it is C4. And in case of C3 convertase in case of classical pathway is C14B to A bar. C3B capital B small b is alternate pathway. C3 convertase in case of alternate pathway. Here MBL or MASP C42B A is C3 convertase in case of lactin pathway. C5 convertase is a C3 convertase plus C3B equals to C5 convertase. Here C5 convertase is C14B2A 3B and C14B2A 3B bar, and here alternate pathway C5 convertage is C3B capital B small b 3B is C5 convertage, here MBL MSP C4B2A 3B is C5 convertage. These C5 convertage convert C5 complement to C5A and C5B. Complement level in the serum means since all the complement participate in classical pathway, so all complement concentration in serum get decreased or low. In case of alternate pathway, C14 and C2 complement are not participated, so their level is normal. Other complement which take part in alternate pathway, their level get reduced or get low. In case of lactin pathway, only C1 complement not participate, so C1 get remain in normal concentration, other complement become low. Classical pathway of complement activation seen in acquired immunity, while alternate and lactin pathway is seen mainly in innate immunity. Now we will see what is the function of complement pathway. First is target cell lysis by membrane attack complex formation. Membrane attack complex may pore or channel in the target cell membrane. Pre-passes of various iron and water into the cell, leading to swelling and lysis and death of the cell. Inflammatory response. Complement byproduct like C3A, C4B, C3A, C4A, C5A are called NF-allotoxin, bind to surface receptor of mast cell and induce their degranulation, leading to release of various inflammatory mediators like histamine, serotonin, and prostaglandin, which produce inflammatory response. Opsonization. C3B and C4B act as major opsonins and coat immune complex or particulate antigen and help in phagocytosis. These opsonins make the immune antigen or immune complex testy for the phagocytic cell, so easily get phagocytosed. Phagocytic cell express complement receptor CR1, CR2, CR1, CR3, CR4 for complement component. C5A augment this process by enhancing the CR1 expression on phagocyte by tenfold. Augmentation means C5A जो होता है ऑप्सोनाइजेशन की प्रोसेस को काफ़ी बढ़ा देता है, तेज कर देता है, जैसा कि यहाँ पे लिखा है कि 10 फोल्ड, मतलब C5A जो है फैगोसाइटोसिस को 10 गुना बढ़ा देता है. Removing the immune complexes. C3B play an important role in removing immune complexes from blood. C3B bind immune complexes are recognized by complement receptor CR1 present on RBC, taking immune complexes to liver and spleen where they get phagocytosed by phagocytic cells. Thus help in clearing blood from these debris of immune complex. Function is in case of viral neutralization. There are many ways to neutralize. Viral by complement with the help of complement. First is complement get coated on viral surface, neutralize the viral infectivity by blocking their attachment site. Second is C3B mediated opsonization of viral particle. Third is lysis the lysis of the enveloped virus either by activation of classical pathway, most in case of most viruses, or sometimes by alternate or lacking pathway. Example, by example seen in like Epstein-Barr virus, rubella viruses. Thank you for watching. Subscribe my channel for further upcoming videos. आने वाले वीडियो हमारे चैनल को सब्सक्राइब करें और जिससे आपको आने वाले और वीडियो के बारे में जानकारी मिलती रहे। थैंक्स फॉर वॉचिंग।